Is my screen visible? Okay. 1 second. Okay, so good evening, everyone. Sorry for the shift, reschedulement of the class. I was not feeling well for a while. So today we'll be discussing week 6, which mostly involves the beginning of glycolysis part and then the bioenergetics part. Then we'll be discussing few concepts first, and then we'll be discussing a few concepts, and then later we'll solve some questions based on it. So let's begin with this. Okay. So first thing is so so we'll be starting with glycolysis. So can anyone tell me what's glycolysis? Hello. You guys can answer in the chat box as well if you want. So I was asking what is glycolysis? So it's basically breakdown of glucose to pyruvate. There is a multi-step process, and where we can get converted to pyruvate. And in this, during the conversion, energy is released, right? In the form of what are the in which form the energy is released? ATP, and one more it's not in the form of energy. Later it will be converted into energy. So there is one more molecule, NADH. Yeah. And so this is glycolysis. Glycolysis is basically the breakdown of glucose to pyruvate. So can anyone tell me in which cells the glycolysis occurs, or maybe any cell where glycolysis doesn't work? No, like like I understand that glycolysis occur in cytosol. Like like is there any cell or any place in the body where glycolysis doesn't happen? So is there any cell or any part of our body, like anything that where glycolysis doesn't work? Anyone? Actually, like as I always as we know that glycolysis is basically the breakdown of glucose to pyruvate, and then which in turn gives us energy. So uh, the answer is like wrong. Actually, all the cells in our body, uh, like in every cell of our body, glycolysis occurs. So that's the thing. Like is that like there is no exception to it. If all the cells perform glycolysis. So this thing is performed by all cells. All cells will perform glycolysis. So every cell we needs energy and everything. So every other cell in our body will perform glycolysis. Now this glycolysis is converted to goes to electron transport chain, and then we get more energy, right? So so what are the two main points like requirement for this process to occur? Like what does the cell need? What does the cell need to perform this electron transport chain? From this pyruvate, it goes to electron transport chain and we get a lot of energy. So what does the cell need? What is the basic two things that the cell need in order to perform uh, this reaction? Anyone? PDH. No, it's not PDH. So like I'm talking in terms of at the cellular level, not at the enzymatic level or something. I'm talking at the cellular level. Every cell. So we know that every cell will have to perform glycolysis. That's for sure. Like all cells perform glycolysis, but not all cells undergo this TCA cycle. So what is the need of the cell? What where does the TCA cycle occur? Yeah, that I can frame that thing. Like where does the ETC cycle occur? Like where does this electron transport chain occur? That might be an answer. Where does this mitochondria? So yeah. So like first thing the like for to convert this pyruvate in order to get into this ETC and to get ATP, we need mitochondria first. Okay, this is the first requirement. So what is the second requirement? First tell me this, like the conversion of glycolysis to pyruvate and then to this ETC. What is it called? Like this complete process, what it is called? Something called aerobic something? Aerobic respiration, right? So if it's aerobic and this thing, then the second requirement is oxygen. So now tell me, like since like glycolysis is happening like everywhere, like like glycolysis is happening in all cells. So is there any cell in our body where this pyruvate is not converted into ATP? Like so like what is a cell where this pyruvate is not there? Like like this pyruvate is not converted into ATP. Is there any cell in our body where this process happen, or like is it similar to glycolysis that every other cell will perform this TCA cycle? Is there any cell in our body where glycolysis does happen? Because we already know that our cell requires ATP and everything. So glycolysis is a must process. So is there any other cell, any cell in our body where this pyruvate is not converted to, like it does not go into TCA cycle and we don't get that much of energy? See, the clue is here: mitochondria and oxygen. So now you guys have to think: is there any cell where either oxygen is absent or mitochondria is absent? So is there any cell where these two are like either there is no mitochondria or there is no oxygen or there is both, like both of them are not present? Is there any cell in our body? RBCs, right? RBCs don't have mitochondria. They have lot of oxygen, but they don't have. Yeah, like that's what, like that's what, like to convert pyruvate, to convert pyruvate into, like to, like ATP, like I mean in the TCA cycle, to go let this get into TCA cycle, we need mitochondria plus oxygen both. But if there is no mitochondria, then like alone oxygen can't won't help this pyruvate to go into that electron transport chain. That is TCA cycle. So we need both mitochondria and oxygen. And RBCs are such cells which don't have mitochondria, but they have abundance of oxygen. They have abundance of oxygen, but no mitochondria, and therefore only glycolysis is limited to it. Only glycolysis occurs, but no TCA cycle occurs. So that's an exception of this aerobic respiration. That glycolysis occurs in every other cell in our body, but this conversion of pyruvate into this conversion of pyruvate getting into TCA cycle and then to ATP is optional. Like optional in the sense, like there are cells that they don't perform because they don't have mitochondria in it. So what is the other pathway? Um, we have read that, okay, this is aerobic respiration. What about other pathway? Where does the pyruvate go if there is no oxygen present? Where does this pyruvate go if there is no oxygen present? We have learned about anaerobic respiration. When there is no oxygen, it forms lactic acid, right? So this is the entire, like, so basically either it goes to the aerobic respiration or it can go to anaerobic one. In anaerobic it will form lactic acid, while in aerobic it will form the, like pyruvate goes into ETC and then ATP is formed. And there is the cell, the RBCs, they don't perform TCA, like they don't have the TCA cycle. So this was it. Next, now where do we get this glucose from? Like we understand that glycolysis is occurring. So like what is the source of this glucose? Like where does it come from? Food, yeah. So whatever we eat, it's basically our dietary substance. So like whenever we eat, we get food, and so when we eat food, after the digestion part, so this is the GI tract, that is gastrointestinal tract, GIT, and then from this, all our carbohydrates are broken down into. So like this is our like carbs. So it has to diffuse out. Like after the digestion part, it has to diffuse in. So in what form will it go out? Like one thing we know, it's glucose. Next, What are the other forms via which this carbs can be like come out of the GIT tract after digestion? So we eat complex carbohydrates, carbs, then it is broken down, and then assimilation occurs. And one of the form of this glucose is like, I mean, this carbohydrate is glucose. So the clue here is like glucose is a monomer, so that we can easily use it. So what are the other forms of monomer, like monosaccharides? What are the monosaccharides? Yeah, like yeah. What are the other monosaccharides? Yeah, fructose and one other monosaccharide. That's one of the most common, like one of the most common monosaccharide that we learn about. One is glucose, one is fructose, other is yeah galactose. Galactose, yeah, right. But do we utilize all the three forms? Because we have learned that glycolysis in like we have learned that glycolysis uses only glucose. So like what about this fructose and galactose? Do we use them? So we know that glycolysis uses glucose, but what about these other forms? Like we know that our body assimilates glucose, fructose, and galactose, all the three, but ultimately they are converted to. Sorry, I can't read this. Um, where is the chat box? Yes, Shiva Ranjani, yeah, like if you have any doubt in this thing, you can ask. Yeah, like I think Rahul, it was Rahul. So yeah, ultimately these things are converted to. Ultimately, these two things are converted to glucose. So basically, glucose is the main form that we use, and this fructose and galactose are converted to glucose, and then glycolysis happens. So, so now like first thing, yeah. So after the like digestion part and then assimilation of glucose, fructose, and galactose, this thing goes to liver via the portal vein. There's this portal vein where it transfers. One second, draw it again. So what we read right now was we eat carbohydrates, it gets digested, and then from there we get three things: glucose, galactose, and fructose, which all are monosaccharide. Then this thing via the portal vein. This is GIT tract, GIT, that is gastrointestinal tract, and from this via the portal vein it goes to the liver. So liver has this, like, what are liver cells called? Like, liver is the complete organ. So what is the unit? Like, I mean, what is the liver cells called? They have this a special name, right? Anyone? Yeah, hepatocytes. So via this portal vein, it goes to liver, and there is this hepatocytes present there. Okay. Now, when this glucose goes here, so like uh, this is where glucose is mostly converted. Like you guys, I hope you guys know that glucose is converted into, like, I mean, it is stored here. So when there is like this is basically a master regulator of glucose. So when you are eating like abundance of glucose right now, but it won't, like, from this like from the liver, it goes to our normal circulation. See, circulation. So if we are using a lot of food, like we are eating like abundance of food, like we are taking abundance of carbohydrates, but this liver does not allow this and all of the glucose to directly go into the circulation. And if it does, we will get hyperglycemia. Glycemia is a condition where glucose is very high in our blood. So that's where this liver plays a major role. It helps in controlling it basically controls the regulation of this glucose. So that's the role of the glucose, I'm sorry, the liver. So when there is abundance of glucose, um, it is basically stored. So what is the stored form of glucose? Glycogen, yeah. So in this liver, the glycogen is stored. Glycogen is stored. Okay. Now, so based on the need, so like as I told you that this liver is the most, like, I mean, the master regulator of glucose. So like when we are having like a lot of food, like when we are taking a lot of carbohydrates in one go, but it's still our body tends to maintain the glucose level in our blood. That is because of this liver. It won't allow all of the glucose to go out. And another thing, when you are like starving in a starvation condition, like what if you are not eating, or in between the meals, when you are like feeling hungry, like suddenly your glucose doesn't go down entirely, because this this glycogen is converted back to glucose, and this glucose is released into our normal circulation, blood circulation. And this from blood circulation, from this blood circulation, so from this blood circulation, is it goes to interstitial fluid, and then to cells. So this is like uh, this is the blood capillary, and then the cells come up. So in between these cells there is a gap, and that's the interstitial, like it's where the interstitial fluid is there. So from here only the cells diffuse. So now what we have learned is like from blood circulation to interstitial fluid. Okay, now tell me, this is the blood capillaries, like our capillaries, where the glucose is high in number, and this is then the interstitial fluid, and then comes our cell. This is our cell. So like, how is the glucose moving from this capillaries to our interstitial fluid? Like, is there any like special mechanism, like transporters, or like is it just diffusion, or like do we need other channels, or like anything? So it is due to diffusion. And why is it so? So as suggestion this, okay, as this like he suggested, the glucose is easily transported, like easily exchanged between the this interstitial fluid and capillaries. They don't need any special like no special channels are needed, anything. So like if the like glucose is high here, then it will automatically go to this one, and if it's low, the reverse happens. So it basically jumps here and there, like it basically maintains the homeostasis in between. Now, Glucose can't diffuse from cell. Um, Actually, I'm not able to find the written tab. Stop this. Yeah, I got it. So you were telling glucose can't diffuse from cell membrane. It's not a cell membrane, it's the blood capillaries. It's in the blood, um, not the cells. Right now it's in the blood. It's in the circulation, not the cells. So I'll come up to that thing. Let's show you one second. It is not in the cell right now. Okay. So as Rahul mentioned that glucose can't diffuse directly, but it is in the cells, not the capillaries. So this capillaries and the interstitial fluid, the glucose can easily diffuse here and like easily diffuse. Like if it the based on the concentration gradient, the glucose will always move from higher concentration to lower concentration. But yeah, he was right. When it has to go from this interstitial fluid into the cell, then it can't diffuse because glucose is a like large molecule and it can't easily diffuse in it. So that's where the concept of transporters comes in. Transporters. So now like so like and as can so that's where the concept of transporters come in. So now okay, so here like diffusion happens when it goes from blood capillaries to interstitial fluid. Now from this interstitial fluid, when it has to go into the cell, the glucose has to go into the cell. That's where special channels are used, and that's transporters. So now what are these channels? Can anyone tell me like what are these transporters? Is there any specific name to it or like anything? Okay, first tell me this. Let me take the basics. So diffusion is of two types, right? What are the two types of diffusion? Okay, like that's based on energy. Like it's the first division is based on simple diffusion and facilitated diffusion. That's the first classification. And then this facilitated diffusion is classified based on active and passive. Okay, so now tell me what is simple diffusion and facilitated diffusion. What is simple diffusion? We were talking about diffusion. One is simple, one is facilitated. So simple diffusion is basically what we learned. The like when there is this blood capillaries and then the interstitial fluid, interstitial fluid. So basically this glucose basically diffuse easily. It does not require any channels, transporters. While facilitated diffusion, since the glucose molecule like from this interstitial to cell, since the glucose is very large molecule for the cells, it has to go via a carrier proteins, yeah, these channels are required. So transporters are required here. Now this can be like this is when there is this can be happening by active and passive. Passive basically no energy, no ATP needed, while active is need of energy. Now this can also be ATP or gradient is needed. Now this can happen. Is also divided into primary active transport and secondary active transport. So now can anyone tell me what is the difference between primary active transport and secondary active transport? Anyone? So basically, in layman terms, I can tell you that primary active transport is like it's one of the transport wherein ATP is utilized on the spot itself. Like if there is, for example, just an example, if I'm transporting some molecule, this is a small molecule, and I need to transport it here against the gradient. One more thing about active transport, it's against the gradient. So it requires ATP. So there is a utilization of ATP at the spot itself. So that's the primary active transport. It utilizes the ATP at the site of transport itself. But when we talk about secondary transport, so in this secondary, the utilization of like first thing is the utilization of ATP is not at the site. So if I'm transporting something, a molecule inside, the ATP, it uses the ATP. It uses the ATP, but it does not. What was I saying? Yeah, it uses the ATP, but not at the site. For example, there was another channel where it is transporting something. So it ATP is utilized by some other transport something else, and there is a change in the gradient of some ions. There is a change in this ions gradient, and this change in the ions gradient is used to transport this molecule. So as I mentioned here, in active transport, there is this ATP utilization or gradient. So in secondary active transport, utilization of ATP is happening somewhere else. And because of this utilization of ATP, the primary source is ATP utilization. That's why it's active. But the main force that has led to this movement of this molecule inside the cell or outside the cell is because of the change in the gradient. Gradient can be a proton gradient or any ion gradient change. And that's how it's happening. So that's the thing. So diffusion can be simple or facilitated. um, Simple is basically when there is no requirement of channel, no ATP, there is no channel required. Then facilitated is there is a transporter or carrier protein or channels, anything are there. And then this can be divided into active and passive. Passive is no ATP, no energy required, and active is either it's a use of energy or the gradient created. And it can be divided into primary active and secondary active. Primary active is basically ATP is happening, like ATP utilization at the site of the transport, and while secondary active transport is basically the ATP utilization is somewhere else, but that ATP utilization is creating a gradient in the cell, and which leads to the movement of other ions in. Okay, now one more thing I wanted to ask here was, do you know the difference between channels and carrier proteins? Transporters, we can just turn carrier proteins. And second thing is the selling forward channels. Anyone? I'm mostly clearing the concepts because in glycolysis it's basically the step-by-step reaction. You just have to memorize those parts. So mostly like I framed few questions on those parts. So I just wanted to like give some insight on what actually is happening at the cellular level and not at the the enzymatic level, the pathway. So that's what we are focusing here. So like can anyone tell me like what is the difference between a carrier protein and a channels? Yeah, conformation, like there is one of the major differences, conformation change. Conformation change. It's not conformation, it's conformation. Conformation. So here there is no conformation. Yeah. Anything else? So when we talk about the channels, like when we talk or think about channels, we think of a structure like this, right? And this is the carrier protein. Okay. Now get to channel and channels are there. Yeah. Something, yeah, those there are those are channels basically. They just open. So now if there is this molecule, lot of molecules here, and there is a lot of molecules here. Now tell me which will be faster? Like what? Two, three, four, five, six? Yeah, six, yeah. So in this carrier protein, it is this, and channels, this will be faster. Yeah, it will be faster. And why is it so? So now, as Rahul suggested, the second point comes here. It is fast. I mean, very fast. While this is comparatively slow. So that's the second point. Now, there is no control on movement. Yeah. So once this opens, like all of them can come in directly. If there is like for example something binds here, like initially it was closed but then some molecules come and bind here, any there are different things like ip3 in some kind of signaling and something. So when that molecules comes and bind and this thing opens up, this thing opens, so they can directly move in, all the six molecules can get in easily. But here we know that the carrier protein acts like this, like once this thing will go and bind here, and this there is a change in conformation, so basically it will go in, it will release the molecule, then go back, go in, go back. So in that way it's kind of slow, like I mean quite slow. So that's the thing, there is a conformation change, only one thing or one molecule or two molecules sometimes in some channels, they bind, then there is a conformation change, it will go in, they will release and then come back in, and then again the process occurs. But it doesn't happen the same thing in the channel part, they just open up and all the proteins, like all the molecules go in. So that's the thing, its channels are fast while carrier proteins are slow. Third thing, these channels, they are not a single unit, they are made up of multiple units. So basically they're multimeric units. So it's like this, it will form a circle, but it's a combination of unit. So like it's a it's not monomeric, it's what is the opposite of monomeric, like it has multiple subunits, like I forgot, it has multiple subunits. It's not a single channel, but carrier proteins, they are mostly a single unit, it's a monomeric unit. So that's one of the difference. Now it is like here it is a fast movement, even if there is like hundreds of molecules it will just move, like it will release it. But here the process will be slow even if the molecules are high, so it will be a process like one by one process. So uh, what is the like capacity then, like if I tell you which will be saturated first, so which will be like saturation is basically a concept where like uh, the protein or enzyme has come to its limit. So of these two, like which do you think will saturate first? Yeah, the concept was simple. Like as you like, yeah, carrier protein, yeah, carrier will saturate fast. This is saturable. Like very, it gets saturated very fast, very fast, because it has to do a lot of work. It has to move from one conformation to other conformation, everything. But here, it does not it does not have. But here it does not have anything like that. It is like it just opens up and then a lot of molecules moves in until and unless this signaling molecules is removed or something happens. So it is not that much saturable, not saturable. So this is the major difference between transporters. So transporters is basically used in facilitated diffusion where there is a need of this kind of channels in order to move mostly large molecules or like ionic molecules or something like that. So it can be of two types, that is carrier proteins or channels. And then this is the example of carrier proteins where there is a conformation chain. Once the molecule goes and bind to this protein, then there is a conformation chain. It releases the protein and then it goes back to the original form. And then again the same process happens again and again. While in channels, it does not. It's just a uh, it's basically a canal type of thing. Once it gets activated, all the things will move in. And this thing, the process is quite slow because it has to do a lot of work, while this process is like immensely fast. And third thing, it's this is a monomeric units, while this is a multiple, like there are like multiple subunits, different channels will have different like subunits in it. And fourth is it's saturable fast, like because it has to do like a lot of conformation change, a lot of energy utilization. It is getting saturated fast, while channels does not. So this was about the transporters. Now we were discussing about we were discussing about transporters in glucose. So glucose. So glucose is transported in the inside the cell via special type of channels. So like I was asking, like what are these channels? Anyone? This is interstitial fluid. Why? This thing is happening by facilitated diffusion. Anyone? Can anyone name these channels which transports glucose? So these channels are called glucose transporters. That's the basic name. In terms, GLUT. Yeah, GLUTs, GLUTs, anything. So GLU stands for glucose, T stands for transporters. So this is glucose transporters. Anyone know how many types of glucose transporters are there in our body? There are about 14 types of glucose transporters in our body. Okay, now like we had discussed about something what FD, like that is facilitated diffusion. So it's either of active type or passive. Now tell me, like glucose transporters comes under which thing? Whether it's active or it's passive. Hmm. Any other guess? Like how many of you think that it's active? You guys can raise your hands if you want. I don't know how to generate a poll in this thing yet, but let's see. Maybe tomorrow if there is one more class, we'll continue this thing further. So then, hmm. It's not active process. It's not an active process. Active process is basically need of energy, and this glycolysis itself is a process of generation of ATP. So what if what if you're taking in glucose, like you're eating a lot of glucose, then your liver is storing something and releasing something in a blood circulation, and then it has to go to your body, but then you're dehydrated, like you have not fed like for a while, like you don't have food, there is no energy in your body. Like obviously that will be a minimal basal amount of energy, but what like it can't be used in order to like it mostly goes to your brain, like it has to help that. So how do you think, like if there is no energy in your body, then how do you think this glucose will be utilized? So that's where nature comes in, and it has played a like very major role in this thing, and then it has made all the glucose thing passive. It does not require any energy to transport glucose from uh, uh, interstitial fluid to into the cells. It does not. It's a passive process. So that's the thing. Okay, it's a passive process. So what we learned here, that glucose is transported into the cell via passive diffusion, passive facilitated diffusion, and then the special transporters are called glucose transporters. In terms, they are called GLUTs, and there are 14 types of GLUTs. But the major role that is played by the GLUTs is 1, 2, 3, 4, 5, and 7, and rest are like not that obvious. Like I mean, like not they do not play that major role. So now we'll be studying about the GLUTs. Glucose, like glucose 1 to like, am I missing something here to tell you guys about? Okay, I've told you the conversion, I've told you the okay, yeah. So we'll be focusing on the glutes. So there are like 14 types of glutes, and then major one is 1, 2, 3, 4, 5, 7. Okay. Now these glutes are present on different organs. Like I mean, like it's not that all of them are present all in every part of our cell, like in all the cells. It does not work like that. It's based on the function. They are present in numerous people, based on their function they are present in different cell types. So like let's start with 1 and 3. Okay, not 2, 1 and 3. 1 and 3, these are mostly ubiquitous, irrespective they are present in every other cell in our body. So mostly like it's in the RBCs, it's erythrocytes, and then the brain cells. So they are like they have the like I mean it is ubiquitous, but these two cells have um, higher number of expression of this these glutes. So basically if this RBC, so glut 1 and glut 3 is present in abundance, so it has abundance in number, this brain and cells. Okay, uh, now these are in abundance of cells. Okay, okay, and one more thing, we all know that at the end of the day whatever we eat, like it's the brain that has to function properly, and then and having this glut 1 and 3 in abundance has plays a very major role in this thing. So like for example, first I'll tell about other glutes too. Now glut 1 and 3, now then let's talk about glut 4. So you guys know that like glucose transport has a like major role, like I mean insulin plays a major role in this glucose, right? Yes, right? Like I mean, anyone? So insulin plays a major role in storage, like I mean the movement of glucose. So when we eat anything, then the insulin is secreted out because it is like when there is higher amount of glucose in our blood, insulin is released, and then insulin goes and binds to certain receptors, and then it sends a signal, and then goes in. So this is our blood capillary. From the blood, glucose goes comes out. Now when there is higher number of glucose, and then this is the cell. So this glucose is sensed by something. Which is the organ that senses this glucose? Like my blood has a lot of glucose. Which how does the body know that it has a lot of glucose? Okay, let me frame the question this way. Which is the organ in our body which releases insulin? Insulin release, like, uh, yeah, I don't like who was that who told spleen? Like, that's entirely wrong. So it's the islets of Langerhans, that is the pancreas. So this glucose is sensed by those cells, and then pancreas, and the insulin is released. And the insulin is released. Now this insulin has to go and bind to the cells. There will be insulin receptor here, and this thing will go and bind here, and then this will signal the cells. Like there will be signaling happening, and then this glucose comes in. Okay. So okay. So like when there is a like higher amount of glucose in the blood, this is sensed by our pancreas, and they release insulin into the blood, and this insulin goes and bind to the certain cells in our body, and then the glucose is taken in. So first thing is this glucose has to get taken into the cell. So this thing GLUT4 plays the role here. GLUT4. So so now one thing that we have confirmed here is that GLUT4 is under the influence of insulin. But we have studied about GLUT1 and 3. It is not. It is ubiquitously present. Like there will be like there will be GLUT1, GLUT, GLUT3, GLUT3, GLUT1. So like in this cell, it will as we have already discussed, GLUT1 and GLUT3 are ubiquitously present, but they are in abundance in number in erythrocytes and brain cells. But they are not in the under the influence of insulin. So like now here a question, like first I'll like finish this thing up first. For example, this so like GLUT4 is under the influence of insulin. So when there is higher amount of glucose, this insulin is released, insulin goes and binds in, and then glucose is taken up by GLUT4. So this is GLUT4. GLUT4. Okay. So now what are these cells where GLUT4 is present? And GLUT4 is under the action of insulin. Can anyone tell me like these are the cells like where the glucose is stored in abundance, like I mean like a lot, in a lot. Okay, yeah. So these are basically hepatocytes because it's liver. Skeletal muscle cells. Okay, and one more, like there's one more organ where this glucose is like stored a lot, like mostly these are the sites of glucose. So one it's hepatocytes, that is liver. Other is like skeletal muscle cells, that is muscles. Mostly like in muscles, like it's not the cardiac muscle or something, it's the skeletal muscle cells. Skeletal muscle cells. So they are the responsible for storing abundance of glucose in it. One more cell type is there. Like when I talk about the energy sources in your body, what does what are the things that comes up in your head? What are the energy sources? One is carbohydrates. Okay, carbohydrates are stored in the form of glycogen. There is one more major source, like it has much more energy than carbohydrates. Fats, yeah. So like another tissue is adipose tissue, or like when we talk about cells, it's adipocytes. So these are the main cells where GLUT4 is present. So I'll clean this area first. So we were talking about GLUTs 1, 3. They are present in you. They are ubiquitously expressed. Ubiquitous. They are mostly like in abundance in RBCs and brain cells, neurons. That is brain cells. When we talked about GLUT4, like first one of the major and most differential um, difference between this GLUT is it is insulin dependent, and this like when there is like a lot of glucose in your body, it is sensed by pancreas. Pancreas release insulin. Insulin goes and activates the receptor, and these glutes are found on the cell, and then a lot of glucose is taken. So basically, when there is like abundance of glucose in your blood, this insulin is getting activated, and this takes lot of glucose in it, like lot of glucose. So these cells are basically skeletal muscle cells, adipocytes, adipocytes, and then liver cells as well. But mostly these are the two forms of storage. Like not even the liver. Most of the glucose when it's in the circulation, like the glucose passes the liver cells and then goes to blood circulation. Like from mostly like I hope you guys remember this liver, then blood circulation. Liver is where it is letting it out, all the glucose, like whatever it comes up. Okay, so what we were talking about, yeah. So liver, whatever the glucose is released out by liver, it goes into blood circulation, and that's where from blood circulation it goes into different cells, and that's where here glucose GLUTs comes in, and mostly GLUT4 and SKM and adipocytes. It's a major site of storage. When it's releasing from here, then only it goes here. So this is the major sites of storage of glucose when there is like abundance of glucose in your body. So that's the thing. So it's the GLUT4. Now, okay. So as I told you, so okay, when there is like lot of glucose in your body, this GLUT4 comes into play. I understand that. So now, like, what if there are like no like less glucose in your body, like very less glucose in your body? What happens then? For example, let me give you an example. You are in a fasting state, and this is a cell, and there is this. First, let's think this is not hepatocytes, myocytes, like SKM, like myocytes. The cells are called myocytes here, myocytes, and there is one more cell here. Think it as RBCs. And as I told you that uh, RBCs have GLUT 1 and 3, and myocyte will have GLUT 4. Now the concentration of glucose right now, like you guys, like for example, someone like you haven't eaten for a while, like it's been like more than 24 hours, and then the glucose amount is very low in your body. Of course, like the glycogen in your body will be released out. But now, which do you think will take up the glucose? Whether it will be GLUT 1 and 3 or whether it will be GLUT 4? Take a wild guess, like anything. You say GLUT4. Okay. Anyone else? When there is a like really less amount of glucose in your blood, which do you think which glut will take the glucose, the minimal glucose? Okay, let me tell you this thing. Amardeep, okay. So yeah, Amardeep is right. Yeah, it's glut 1 and 3. First thing, let's go by logic first. Then we'll talk about the nature's play. Okay, first thing is like glucose is quite less. First thing that you have to know that GLUT4, what 1 and 3 cause brain function is important. Okay, like going in that way. Okay, like you can think that way. Yeah, sure. But let's talk about logic first. First thing I told you that GLUT4 is getting activated based on insulin. So that's the thing. That's a golden rule. Like GLUT4 is getting activated by insulin. I'll tell you about this pathway, right? Like after this, like how it is getting activated, so that you guys can get a clear idea of what's happening inside the cell. So like first thing is that okay, now GLUT4 is activated by insulin. Now one more thing I mentioned that it's the fasting state. And what does the fasting state mean? Low glucose. And when there is low glucose in the blood, why do you think insulin will released? Like will there be any insulin? Like I mean if already there is glucose less, then what is the role of insulin here? So do you think insulin will be released? Anyone? Hello, like am I audible? Yeah. So insulin won't be released, and if there is no insulin, then why do you think the GLUT4 is getting activated? So GLUT4 won't be activated. So that's one role. So even though GLUT4 has the it can transport like higher number of glucose. So like for example, if there is a lot of glucose in the body, like abundance right now, if you're like have eaten a lot, like a lot of glucose in the body, then if you're saying that GLUT4 is getting a lot of taking in, I understand that because a lot of glucose means insulin will go and activate this, and lot of glucose will come in compared to this one. Like for example, if only one molecule goes here, like five molecules will come here. So that's the like capacity of it, like it can take in much more, like much of it. But the affinity of glucose, affinity of glucose to this GLUT1 and 3 is very high compared to this GLUT4. So one thing point I am mentioning here is affinity of glucose. Affinity, basically affinity. 1 second. Hello, like am I audible? Hello, am I audible? Yes, okay. So like we were talking about glut. Okay, so we were talking about the affinity part, right? So the affinity of the glucose is higher for GLUT1 and 3. So affinity of the glucose is higher for 1 and 3 compared to GLUT4. GLUT4 can take lot of glucose in when it is activated by insulin, but the affinity of glucose is higher for 1 and 3. And then now what does that mean? One second. Sorry, like my system didn't have charge. Okay. Uh, so like we were discussing about okay, like can anyone confirm that I'm audible? Sorry for the delay. Am I audible? Okay. So like we were telling you that yeah, glucose 4, GLUT4 will take lot of GLUT4 takes lot of the GLUT4 takes lot of it takes lot of glucose in, but the affinity of the GLUT1 and 3 is very high. Now what does that mean? What does the affinity mean? So now affinity means basically uh, in layman terms I can say that the love of glucose for GLUT1 and 3 is very high, that even if in the minimal concentration it will take up in. So for example, if the glucose is like very low in our body, so it will be easily taken up by it will be easily taken up by 1 and 3, but it won't be taken up by any other, whether it be glucose, GLUT4, 2, anyone. Like it will be easily taken even in the basal quantity, even in the basal amount. When the glucose is very low, it can be easily taken up by GLUT 1 and 3. That is by our brain and RBC. And like um, GLUT4 is not present on these cells because like this process can't be dependent on like I mean like for this, at least the brain cells and RBCs, this process can't be dependent on the insulin. For example, there are like multiple conditions where our cells get inactive, like insensitive to insensitive to insulin, where there is like that's the like metabolic syndrome where diabetes comes in. So where like either the cells don't have the receptor for the insulin, or whether the cells have grown insensitive to insulin. That is like even if the cell like insulin is in the like larger number, the cells can't pick up the insulin, like they are not in they become insensitive, or sometimes the insulin is not produced. So this, like for at least for our brain and RBCs, this process of glucose intake can't be dependent upon the insulin thing. That's why GLUT4 is not present on the brain or RBC, brain cells or RBCs. So that's one of the major role. And even if your like body is in fasting condition, if there is like you don't have any energy, like if you don't have any glucose, you're still your brain needs to function. Your RBC blood cells need to carry the glucose. They are like like they're really important, and that's why the nature comes in, and that's why glut 1 and 3 has the higher affinity to this glucose, even at the basal level, even at the minimal level, and it is not dependent on anything like insulin or anything. So that's one of the like very important parameter here. So what we have learned is glut 1 and 3 is ubiquitous. It is present on RBCs and brain cells in higher number. And then what's the other thing we'll have learned? Yeah, ubiquitous RBC and brain cells higher number, and it has higher affinity. So this is the term you have to remember always. It has a higher affinity to glucose compared to any other gluts, any other compared to any other glutes. And then second thing, even at the basal level, it can take up glucose inside. So that's the first point. And then glut 4, what we have learned about glut 4, this is also one of the special glutes. This is like it is insulin dependent. First of all, it can take a lot of glucose. It can take a lot of glucose once it gets activated, taken. Now, insulin dependent, lot of glucose taken. Next is what else? It is present on SKM, that is skeletal muscle cells, and adipocytes mostly, because this is where the glucose comes in after the blood, like from the blood. So this is the major points. Okay. Now let's go to glut. So these are basically one-way transfers. Like from this blood cell, the glucose goes just one way. So there is one more special glucose glut that does both the ways. It can go from here to here or like from here, like in the same via the same channel. Well, except this glut, all the glutes just transport the glucose is only one way. So can anyone like take a guess? Like we have 1, 2, 3, 4, 5, 7. Anyone can take a wild guess like which glut can be the two-way transport, or like it can transport two ways. Glut 2, 5, and 7. No, just um, it's just one glut. It's just the one glut. And when like it might be obvious, I guess. Still, it's glut 2. Glut 2, like like you can remember it easily. Glut 2 does both the ways, two-way transporters. Okay, it does two-way transporter. So now, if it does two-way transporter, where can it be present? Where can this thing like play a very crucial role if it's a two-way transporter? Liver, that's right. Liver. Any other place? First thing I wanted to show, like talk about this the mechanism of glut 4, like once like it is like one of the important side. Okay, let me shift it here. Don't have a different slide, I guess. Okay, like I'll just teach you this. Like this glut 4 is like one of the major, like I just want you to so that you guys can imagine easily. uh, okay, for example, like if this is the pancreas, just think this as a pancreas. about how pancreas looks. Something like this. Pancreas, it releases insulin. Okay, and this is a, like, for example, we'll take a SKM, skeletal muscle cells, and this is a receptor. Receptors are basically like these are basically protein itself, but once they are like responsible for sending the signal, like extracellular signal inside the inside into the cell. So it's basically a form of communication here. So insulin, when insulin is released, it goes and binds to this insulin receptor, and from the insulin receptor, like these cells have special vesicles in it. These cells have special vesicles in it. Do you think this is a vesicle? And on this vesicles, glutes are present. So now, when there is this like higher number of, like, I mean, now think this is a blood and glucose is very high. So when this is sensed by this thing, it releases insulin, and insulin goes and binds to this insulin receptor, and this there is this pathway, like, I mean, like lot of, like, this is a, like quite a big pathway, and this thing, because of this activation, there is this some G proteins here. Like, I'm not going into that detail. Let it be. So like when this thing gets activated, these vesicles are transported towards the vascular membrane. Sorry, not vascular, plasma membrane, and where they fuse. So basically they get like this. So once the insulin gets bound to it, they get fused with the plasma membrane, and that's when they're like they're the found. So like they are not always present on the plasma membrane, but when there is like high amount of glucose into the blood, this insulin is secreted out. This it is activated by, like, it goes and binds to the insulin receptor, and these vesicles now, like this, they are basically think it as in inactive form, and when the signaling comes in, they go and bind to the, like, they are basically not bind, fuse with the membrane. These vesicles fuse with the plasma membrane, and then the glutes are outside, and then that's how the glucose goes inside. So this is how, like, it occurs. Like, this is the insulin dependent pathway of GLUT4. So it is initially, they are not always on the membrane, while in RBCs or brain cells, the GLUT1 and 3 are always present. So they are always present, but they are, like, in GLUT4 it's not always present. Once it gets activated by the insulin, it goes and fuses with the plasma membrane, and then it is expressed there. So that's the mechanism of GLUT4. Okay, now, so this thing you have to remember. That's the function. Now we were talking about liver, like GLUT2. GLUT2 is basically a two-way transporter, like two-way transport. Like, first organ it's liver. Second thing, it's pancreas. Pancreas have two-way glucose, so that it can easily sense. Like, as I told you, this pancreas, they sense this glucose change, right? But then only they will release the insulin. So this glucose, to in order to sense it, it has to take in, right? So this will go in. So this thing here is GLUT2, and some amount of glucose is also released by pancreas. So that's why it's two-way. So it will take in, like, if there is a change in the glucose in the this place, it will go inside the pancreas and it will come out. This when it is sensed, the insulin is released, and then this GLUT4 is getting activated. So the second thing is pancreas. Um, okay, am I forgetting some other organ? Hmm, I think I'm forgetting something. I think there's one more. I don't remember that action exactly, but okay. Okay. Maybe I forgot. Like, if I have forgotten, then I'll tell you that in other class. Like, we will continue this tomorrow as well, same timing. If you guys are interested, like, we can discuss further. Uh, so here, like, it's a pancreas. So now, liver. Let's talk about. Okay, Here on. So we were talking about GLUT2, GLUT2, and it's present in liver and pancreas. The pancreas, the role of the GLUT2 is sensing, mostly sensing the change of glucose. Now in liver, so as I told you, like when we eat food and then when the GIT tract, that is a gastrointestinal tract, that is GIT, gastrointestinal tract, it gives out glucose, and this glucose, like plus fructose and galactose as well, this goes to liver. So liver, so this is the liver side, like this is a liver cell, hepatocyte. They have GLUT2. When there's like abundance of glucose in, so inside our, like I mean, via the portal, when it comes in, it takes it in and store it in the form of glycogen, right? Glycogen. Even this glucose and galactose are converted to glucose, sorry, fructose and galactose, and it again is stored in the glycogen. Now, when we need, when like you have in the fasting state, or like when you have not eaten in a while, so that in that situation, what happens? This glycogen will be broken down into glucose, and then it will be released out again in the form of, like I mean, like we have this GLUT2. So this is GLUT2. So what is this process? Like, can anyone tell me, like, what is this process of like glycogen broken down into glucose? Like, can anyone like name this process? Gluconeogenesis. Okay, your Papin is right. It's not gluconeogenesis. It's glycogenolysis. This glycogen itself is a form of glucose. While when we talk about gluconeogenesis, sorry about my handwriting, it's gluconeogenesis. When we talk about what is gluconeogenesis, it's obviously like it's formation of glucose. But from what is the um, what do we call, what is the raw material for it? Yeah, for glucose neogenesis. Anyone? Yeah, glucose from non-carbohydrate sources. And that's like, what is the example of that? Like I mean, like non-carbohydrate, for like, what is that? What are non-carbohydrate sources? It can be protein, it can be from fats or glycerols, something like that. Glycerols. So these are non-carbohydrate sources, and when from this glucose is formed, it is glucose neogenesis. So like, what are the sites of gluconeogenesis? Where in the body gluconeogenesis occur? Where in the body gluconeogenesis occurs? last thing is kidneys. Kidney is a site where gluconeogenesis occurs. From there, like I mean, it is converted like from like lot of sources. Like I mean, like what before it getting excluded out, like this is the source, one of the source, like where gluconeogenesis occurs. And there is one more tissue, two more tissue. Like there are three. I guess I forgot about this. I think I forgot about this gluconeogenesis. So like I mean there are two more sites where this gluconeogenesis occurs. So like okay, we were discussing about GLUT. So like that's where like gluconeogenesis occurs. Okay. Now so we have read about GLUT2. GLUT2 is basically present on liver and pancreas. In pancreas, the main role is sensing the change in glucose, and based on that, it will release insulin, and gluts are GLUT4 is released out in liver. Uh, its main role is like I mean based on the requirement of glucose that we need, it is either goes in or whether it goes out. So that's another thing, and it is a site where glycogenolysis occurs, and there is this term called gluconeogenesis as well. Now we have discussed out 1, 2, 3, and 4 as well. So what was the specialty? Like 1 and 3, like let's get a recap. 1 and 3 are present ubiquitous and most abundant in RBCs and neuronal cells, neurons, and they are like quite special because like their affinity is very very high. So even at the basal level, when we are like I mean very fasting state, when we are in fasting state and anything, it will still take up the glucose in order to feed the RBCs and the neurons. Then there is this GLUT2. The specialty of GLUT2 is it can transport both sides, like it can get in and get out as well, and it is mostly present on liver and pancreas. In pancreas, the role was to sense. Then in liver, it was like from glucose to glycogen and glycogen to glucose based on our needs. And the last, like the GLUT4, the specialty is insulin dependent alone because it's the only GLUT that is insulin dependent, and it can take a lot of glucose in, present on skeletal muscles and what is the other thing? Adipose tissue. Adipose. Okay, now we have come down to 5 and 7. 5 and 7. Gluconeogenesis also occurs on in liver as well. Um, there is one more organ as well. I don't know it's whether it's at the spleen or I don't remember. I think it's the spleen. I think it's the spleen. Okay, it just says liver and kidney. I think it was split. It's not the spleen, I think, but I remember it was 3 or I forgot maybe. Okay, now we'll go with 5 and 7. GLUT5 and GLUT7. So now there's this special thing about GLUT5 as well. So unlike all the glutes that they transfer glucose, this GLUT5 does not transfer glucose. It transfers fructose. So that's this. It's is it? It's like this is its specialty. Fructose. It transports fructose. Okay. Now, like just a random question, like which is sweeter, like fructose or glucose? Fructose is more. Okay. Okay. Now this fructose, this GLUT4 is present, which transports fructose, and in our body, like there's this cell that is like highly, like I mean, it has fructose. Um, can anyone like guess, like I mean, which cell in our body is like I mean, like the amount of fructose is quite high. It requires like fructose, like it requires fructose or like the sweetened things. Anyone? Which cell in our body takes up fructose? Like I mean, the content of fructose is quite high in that cell. So I think like it's one of the facts that our sperm cells have a lot of fructose, and then the producer. So like these cells have GLUT5 in it. The sperm and testes, like they are like high in fructose, and that's why the GLUT5 takes in. Like I mean, GLUT5 is also present on the GI tract, as I told you that uh, from GI tract glucose comes in, fructose comes in. So here also GLUT5 is present on GI tract as well. And then GLUT7, this is the last interesting one, that is it is present on endoplasmic reticulum. When gluconeogenesis occurs, when I tell that where the like the gluconeogenesis occur, it mostly takes up in like endoplasmic reticulum, that is SCR, smooth endoplasmic reticulum. So that's where like it transports the glucose here. So we have covered all the glutes. So like a last summary, like I want you guys to answer 3, 4, 5, 7. So first thing, like let's like a pop quiz type of thing. First thing is, yeah, glucose transport. Is it active or passive? No, like first, like let's go up. Like whether it is done by facilitated diffusion or like simple diffusion. Glucose is transferred via facilitated diffusion or simple diffusion. Am I audible? Facilitated diffusion. So yeah. So first thing was about the glucose was facilitated diffusion. Facilitated. Okay. Next, it is either by active transport or passive transport. It's passive. Okay. So basically, no need of ATP. So that's one question. Okay, like one more thing I wanted to ask. Okay, like I'll ask after this. Okay. So that's the thing. Okay. Now this is only a few things that I wanted to ask. Right? Glucose. Yeah. So like when the glucose is stored in the form of glycogen. Okay. So uh, so what is the process of glycogenolysis? What is it? Like, what is the breakdown of in what? Like what is it? Glycogenolysis. The term of gluconeogenesis. So what is glycogenolysis? Glycogen to glucose. Okay, yeah. And gluconeogenesis is basically from non-carbohydrate source to uh, glucose. Okay. Now, GLUT1. First, like which is the GLUT that has the higher affinity to the glucose? Higher affinity means like even at the smallest, the basal level concentration, it still takes up glucose. 1 and 3. Okay. Now, which glucose is activated by papain GLUT4? It's not GLUT4. GLUT4 is again Higher affinity means the basically the love for glucose is so high that even at the minimal, like even at the lowest concentration, it can take in. Like there is no requirement of activation, there is no requirement of any movement, there is no insulin activation, no and nothing. So it's a glut 1 and 3. Glut 1 and 3 has the highest affinity to glucose. Like even at the minimal concentration, when we say that uh, any enzyme or anything has a higher affinity to some other molecule, it means that even at the lower concentration, even if you are just giving them 1 nanogram or like 0.5 nanogram, it still gets activated. So that's the highest affinity, that even at the smallest concentration it will go and bind. So that's the glutathione and 3. So now like which glut gets activated by insulin? Okay, that's GLUT4, insulin mediated. Okay, now which is does the two-way transfer? Two-way. Okay, fructose transporter. You can remember this by GLUT5. 5 means fructose. So that's fructose, right? And this is okay. Now we'll talk about um, which is ubiquitously present. Like every other cell will have this two glutes. So ubiquitous, like even this like for example this GLUT4 is present on SKM and adipose. Even this cells will have your 1 and 3. Even this cells will have 1 and 3. Every other cell will have 1 and 3, which is present on pancreas. It is for the sensing of glucose. It's pancreas to sense the glucose, and its liver to like send the glucose based on the requirement. Now, fructose. This is present. GLUT5 is present on sperm and testis. These cells have the highest concentration of fructose. They are quite sweet. And GLUT7 is present on ER, that is SER, where the gluconeogenesis occurs. When I say that liver and kidney cells are the site of gluconeogenesis, I'm basically inside the cell. I'm basically mentioning the smooth endoplasmic reticulum. That's where the this thing is happening. Okay, and this thing are present on RBCs and brain cells, RBCs and brain cells. Okay, we have almost covered everything in the glutes. Okay, and while the rest of the like there are 14 glutes, while the rest of the glutes are like not that special. They're just like normal um, thing. Yeah, the normal one. Okay, now I wanted to ask one thing. Okay, two basic out of the curiosity question. Um, I told you that this is the blood vessels. This is interstitial fluid, and this is the cells. Okay, and this have like transporters in it, and here like there are like glucose coming in. Here is also glucose is there. Glucose is there, and here also glucose is there. And here the glutes are activated. Think this as SKM cells, myocytes. So now tell me, and this is moving from here. It also can move from here. Simple division. Now tell me, if the starting, like this is a hypothetical question, like obviously it won't be possible that the concentration of glucose here and the concentration of glucose here, it's equal. Like let's take this as 1 kg thing glucose. This is always in molar concentration, but I'm not giving that thing. It's just a simple that thing. 1 kg, and this is all kg. So now tell me which glucose will be cleared fast. Whether this thing will go inside first, or whether this thing will go inside fast. Which which side? Let me number this one and type. Which out of these two sides, one and two, which will clear the glucose fastest? Everything is active, like everything is like, I mean, like everything is happening, whatever the process you're thinking, whether you're thinking diffusion, whether you're thinking like simple diffusion, whether you're thinking facilitated diffusion, everything is happening here right now. Like here, like it's the blood capillaries, here it's the SKM cells. So which out of the sides, one or two, which will clear the glucose fastest? Okay, what are the answers? Like, one second, I'll go to the chat box. Okay, so Pepin said neither one nor two. Two from high concentration to low concentration. Naveen Shanmugam also said two. Okay. First thing like I'll answer Papin. First thing like you said that neither one nor two. Think there here like there is no, like there is zero glucose here. Like one more condition is zero glucose, zero glucose. But these two are empty, but there is like equal amount of glucose both here. Like you might think that okay, if there is no glucose here, then how can it be glucose here? Like, that's why I told you it's an hypothetical question. Here the glucose is present. Here also the glucose is present. This is empty. This is empty. And the rest of like I mean the so Papin the movement of phase is that thing. So second thing, I think Amardeep was the one. Mm, So it was Rahul. Okay, Rahul two from high concentration, low concentration. So Rahul, um, similar thing. Like here it's the high concentration, here it's low concentration, here it's high concentration, here it's low concentration. So obviously it will go won't go move this way. Like I mean, it can move this way, but I'm telling the flow is say this way. So I'm asking you guys to think at the cellular level. Like I mean the main level. Okay, let me answer this. So first thing, the answer is two. But the rationale given by you guys, like, I mean, um, Naveen, if you want to give any other rationale, like why do you think it's two? Okay, now I'm telling you the answer is two. Now, can anyone give me the rationale behind it? Why do you think it's two? This can take a while, yes. I think it's fine. Okay, first thing, as I told you, here only simple diffusion is occurring, and this is facilitated one. So I hope you guys might have learned, like read about the graph where the simple diffusion is like something like this, while the facilitated is like this. The rate of clearance of glucose is much higher when there is a involvement of channels. This is simple diffusion, this is facilitated diffusion. So that's one thing. And second thing, it's the SKM cells. Like main of the reason is this simple diffusion can clear like even though the transfer is like even though the transfer needs no extra protein, it can easily diffuse. But the fastest diffusion is facilitated diffusion in all cases. because these channels, once they open, they take abundance of glucose in one go, and mostly this is an SKM cells. So as I told you, SKM cells means GLUT4. Always try to like understand like what you guys are learning. So it's a GLUT4. So already it will take a lot of glucose. There is no glucose that is comparable to this, and then no GLUT that is compatible with this. So when there is the presence of channel protein, it will take a lot of glucose in. So that was the question. Now one more thing I wanted to ask was, okay, I can only take this. So we have till now we have learned that the concentration of glucose is always moving from higher to lower concentration, higher to lower concentration. Like always there is this gradient maintained. The gradient is always maintained in order to move glucose from in and out, in and out. Is there any cell in our body where this like mostly this interstitial fluid and the inside the cells are equal? Like I told you, like this interstitial fluid has higher glucose concentration, then only it will go in. Like always this glucose will move from higher concentration to lower concentration. This gradient is always maintained in order to move this glucose. They are never equal. They just keep moving. Like once they go in, they are like converted to other form or utilized on something like glycolysis occurs. It can never be equal. But is there anywhere in the body where the concentration of glucose in interstitial and the cells are equal, are comparable, anything? We just read about it. We just read about it. Just, I mean, think. It's not hepatocytes. Pancreas, yeah. Naveen is right. Pancreas, because pancreas senses glucose, so it always has to maintain. Yeah, that's right. Yeah, that's the right answer. Yeah. So it's a uh, pancreas. They always have to maintain the glucose equal to the outside glucose. That's when they can sense. So if there is more, they have to get more glucose in so that the insulin is released. If it's less, it has to sense that okay, it's less, then I don't need insulin. So that's the main answer. So it's like glucose thing. Okay. So like time's up, but mostly like I know like we haven't discussed the questions, like mostly it's just the pathways. Like I have made some question, like a lot of questions on that. Tomorrow like I'll brief something about, like I mean like I saw the lectures, like I understand the pathway, but most of the questions, if you guys are planning to do like I mean clearing net or something, it won't be on the pathways itself, but it is based on these concepts. So tomorrow, like on the same timing, that is 7 to 9, same timing. I'll be like I'll be organizing the schedule, and then tomorrow we will complete this glycolysis part, and then we'll solve some questions that I have like arranged, and then I'll take the doubts in terms of those questions. So like if you guys have uh, any other doubt in the things that I've taught you, you guys can answer, like ask. Or we can meet tomorrow if you guys are fine. We'll continue this glycolysis, and tomorrow, like tomorrow, I'll try to speed up the session. Like I think I was taking a lot of time, but I hope you guys understood, and I think this concepts are new to you. So these are the main concepts. Like you guys can memorize all the pathways and all the enzymes and everything. But this is the main concept. Okay, one more thing, like in terms of glycolysis, like where are those enzymes present? Like I mean, the whatever the enzymes we are learning about, hexokinase, fructokinase, or like the conversion and everything. Where are those enzymes present in the cell? Anyone? Just before, yeah, that's the cytosol. It's the place where glycolysis occurs. Okay, then uh, we'll meet tomorrow, and we'll try to take up like continue the session, and then and I hope this session was useful to you. Like we'll solve some questions tomorrow as well, but these are the key concepts. Like you must have like studied all these things. Like I mean, the pathways are like in every other textbook, but these concepts that I taught you like because even at my master's level, I couldn't like I didn't know this stuff. So I think it was interesting for you guys. I hope it was interesting.